Thank you all for having me. Thank you, Dr. Stresser, for inviting me. I'm just going to kind of jump into it. We're going to talk about kidney disease and epigenetics and why we all should be epidemiologists. But before that, I'll talk about kind of my path to epidemiology, just give you a little backstory on my life. So I grew up where that red star is. It's called Lee County, Alabama. If you just get on 280 south, or I guess southeast, and drive about 2 hours, you'll drive right through it and won't even realize that you went through it. And this area called Waverly. And this was really my introduction to health disparities, seeing people have strokes in their 30s and 40s, people having to wait 45 minutes for an ambulance, not having access to healthcare. And so kind of really early on in life, I said, I think I want to do something about this. And I think the best way for me to do that is to become a physician. And so as I got older, I was like, I'm tired of living in this small town. I want to go off to the big city. I want to go to college. So I went to Howard University in Washington, D.C., and really was very green. I just knew to be a pre-med major. And so someone told me to major in biology, and that was just the end of the story. And so kind of pretty early on in college, I got lost in the shuffle and I didn't really know what I wanted to do anymore. And ran into an old family friend who told me, Alana, it really doesn't matter what you major in. Study what you love, take your prereqs, you'll be fine going to med school. And I said, I don't know about that, but sure. So I end up taking just some elective courses in humanities and in the classics department. And I happened upon a class where I learned about the plague of Athens, which was this plague that happened in 427 BC that basically is the beginning of the end of the Athenian Empire. And so I really like this idea of integrating history and culture and society and also medicine and science. And I said, oh, okay, I think I kind of like this. Maybe I should major in this. And so Professor Molly Levine was my professor for this course. And she ended up kind of convincing me to change my major to Latin, which was very strange. And so I was this pre-med Latin major talking about Lucretius and all this stuff during the fall and the spring semester. But in the summers, I was participating in a program called Paradigm here at UAB, where in the summers between sophomore and junior year, I actually came here and I did research in a kind of translational research lab and also was shadowing. And that really exposed me to this process of being a physician scientist, because up until this point, I had no idea what an MD PhD was. And so I kind of ended this Paradigm program and knew that I wanted to do research and that I wanted to be a physician scientist, but I also knew that I really liked the work that I was doing in my kind of Latin studies, thinking about history and society and culture. And I didn't really know how I could blend that into, say, a molecular biology PhD. The other thing was that I really, really hate mice. I'm terrified of them, and I could not work in a research lab with mice all the time. And so I knew I wanted to do research, but I didn't quite know what that looked like. And so come back to Dr. Molly Levine, and she says, by the way, Alana, I happen to live 7 blocks away from the NIH in Bethesda, Maryland. And I also have a fully furnished basement apartment that I like to rent out to my students for cheap after they graduate. They're going to grad school, studying for the bar, things like that. And I think you should come and live in my basement, yes, in her basement, and go do research at the NIH. And I said, okay, sure. I don't know how I'm going to do that. But she says, no worries. My sister is actually a world famous geneticist who discovered the Tay-Sachs disease mutation. And she knows there's this post-bac program that you can do, and we'll help you figure it out. So I happened to kind of meet some folks along the process who helped coach me through the application process, including her sister. And the application process for me was a little bit difficult. I kind of applied to a lot of places, didn't get a lot of bites working with some of those NIH researchers, but actually got all the way to the end of this interview process with someone named Vince Bonham. He's actually the NHGRI deputy director currently, and he's an attorney, he's a lawyer by training. And he was doing the sickle cell research and thinking about genomics and kind of social determinants of health. And I thought, yeah, I think this is the guy I want to work with. So then about a week after college graduation, I'm preparing to move into Molly, or I'm moved into Molly's basement. He calls me and says, hey, Alana, sorry, decided to go with somebody else. And I was like, oh, just a little bummed out. But Molly says, hey, Alana, don't leave, don't go home yet. You're in the basement already, and you can always find a job as a Latin teacher because nobody has a degree in Latin. And she was right. So I started applying for jobs in Latin and as a Latin teacher, and pretty immediately got a bunch of job offers. And so the day before I was supposed to accept a job offer as a middle school Latin teacher, I got a call, or really my parents got a call from this guy, Dr. Kevin Gardner. He accidentally called my emergency contact on my application as opposed to my actual number. And he says, hey, Alana, My name is Kevin. I heard from my friend Vince about you. He said that, you weren't necessarily the best fit for his lab, but that I may be interested in working with you. And so he's like, why don't you come in for an interview? So I come in for my interview, and basically he offers me the position on the spot to research in his lab because Dr. Robin Lorenz, who was then program director of the Paradigm program and then the program director of the MD PhD program here at UAB, had written one of my recommendation letters, and he called her and she said, no, yeah, take Alana right now. And so he offered me the position, and now I'm at the NIH. And I tell that story to just kind of share the role of sponsorship and mentorship in helping me get to this point, and really just being curious and open to new experiences is what led me to this kind of MD PhD space. And so in my project with Dr. Gardner, I was studying breast cancer epigenetics and obesity and metabolic imbalance, and didn't really know what I was doing. I just knew I wasn't working with mice. And he had me doing some data analysis, and I was co-mentored by an epidemiologist there for that project. And I said, oh, wait, I think I want to be an epidemiologist. This is the integration of all the things that I like about research and science and medicine. And so I applied to these MD PhD programs, thankfully got into UAB, and this was the best fit. And I came here because I will say, back in the olden days, no one really understood what an MD PhD epidemiologist was. They didn't see the value in it. And I think now, post 2020, we understand the value of an epidemiologist. So I come to UAB in 2017. I do my kind of preclinical years of med school, just all my classroom work. And then in the middle, you do your PhD science. And as Dr. Urban kind of alluded to, a lot of that was done at home because of COVID. And then I finished my PhD, defended my dissertation in 2023, and I went back and did my clinical rotations. And then, as Dr. as Beth has alluded to, that I will be matching this Friday. And so this morning I found out that I matched. We'll find out where that is. Thank you. Thank you. So just briefly about working with Dr. Irvin, because I think some of you guys should definitely work with her if you have the opportunity to. We're kind of a broadly cardiovascular genetic epidemiology and statistical genetics lab. Basically, we do lots of omics, and anything that falls under kind of the cardiorenal metabolic axis is par for the course. And so these are all the types of data and the specific data endpoints that I specifically worked on, but there are additional things that you can work on. So if you have a question, just wave at her in the back and start a project. And she's a great mentor. So definitely recommend that. So in my dissertation, I wrote my dissertation on genetic and epigenetic risk scores for chronic kidney disease in African Americans. And I'll talk a little bit about what that work was, but I created a polygenic risk score, which is a type of genetic risk score, a methylation risk score, which is a type of epigenetic risk score, and then try to figure out how we can use those risk scores to predict kidney failure. Like I said, about a month after I defended my dissertation, I went back to medical school on a rapid cycle, finished my rotations, applied for internal medicine, and like I said, I'm here now. So that's enough about me. Onto the research. So kidney disease is the 10th leading cause of death in the United States and globally. We also know that more than 1 in 7 adults in the United States have what's called chronic kidney disease. And we think about chronic kidney disease as a disease of the aging. So as we have a population of people who are getting much older, we're going to have more kidney disease. But even when we account for that kind of age factor, we still see increases in the rates of kidney disease, particularly since 2019. And what's particularly sinister about this is that for people who have severe CKD, a huge proportion of people do not even know that they have disease, mostly because CKD is often asymptomatic until you get to those more severe stages, almost to the point of kidney failure. And so because of that, we know that there are more than 800,000 adults in the United States with kidney failure. And this is an independent risk factor for cardiovascular disease. It's an independent risk factor for early mortality as well. And while there are lots of reasons that originate in the kidney, why people have kidney failure, over 50% of kidney failure is actually due to hypertension or high blood pressure and diabetes. And so that's particularly sinister again, because almost half of adults in the United States have hypertension, and most of those folks do not have their blood pressure under control. Similarly, we know that diabetes affects a large percentage of the population as well. And again, not everyone with diabetes has their diabetes under control as well. And you say, well, what does this matter? We have dialysis, we have pig kidney transplants, we can take care of kidney disease. And technically, that is true. However, kidney replacement therapy, like dialysis, really only has about a 5-year survival of rate of 40%. And so if we can prevent people from getting to kidney failure, that should still be our goal, even though we have all these therapeutics for folks once they're at the stage of kidney failure. And so what does omics have to do with any of this? So we know that the heritability of CKD is high. So basically what that means is that while we estimate functions of GFR, which is a type of way of measuring kidney function, similarly that we use blood pressure to determine hypertension, we know that those heritability estimates for GFR range from as far as low as 30% to as high as 75%. So it's really high. And so a huge proportion of that has to do with your genetic background, your DNA sequence. And we know from prior GWAS studies that there are genetic loci that are associated with kidney function. However, the genetic component doesn't fully explain all of that heritability. And this is what we call missing heritability of disease. And so there's this idea that epigenetic modifications, and I'll talk a little bit more about what that is, can account for this quote unquote missing heritability. Kind of the second thing that's happening in the genetic epi space is this idea of polygenic risk scores. And Dr. Armstrong talked to you guys about that a few weeks ago. But essentially, it's saying, how can we take this data that we have, all this genetic data, and apply it into a way that's clinically useful? Because I will say, when you're a physician in a 20-minute clinic appointment, you got to have something that's quick and dirty that you can use to help treat your patient and manage their disease. And so these polygenic risk scores are being developed kind of in mass and for all different types of traits, but we're not seeing similar development in the epigenetic risk prediction space, even though the data suggests that the epigenetics does contribute to disease. And so for us, kind of for this project, that was really where we wanted to kind of anchor this research. And so I created a methylation risk score, but before we even get to that, I just want to make sure we're all on the same page, right? What is methylation and why does it matter? So these epigenetic modifications, of which DNA methylation is a type of epigenetic modification. These are chemical changes that are happening along the DNA strand that can influence gene expression. So it's all just a basic chemical reaction. So a methyl group is a carbon and 3 hydrogens all put together. And depending on where they are and how many there are around the DNA, that can turn genes on and off as far as transcription to RNA and translation to protein. And so we know that methylation changes are dynamic to the environment. And that's to say things like your age, having different metabolic imbalance, things like that can affect how your DNA is methylated. We also know from these studies called EWAS, which are similar to GWAS, they're just looking at methylation, or epigenome-wide association studies, have identified these CPG sites, these locations of methylation that vary in association with kidney function, and in particular, have identified locations that actually causally influence kidney function. And so our idea was, well, we're seeing all this work and development in the polygenic risk score space. There is, a scientific and biological plausibility for the epigenome to be playing a role here too. Why don't we create a methylation risk score? And so why do any of these risk scores matter, right? So the idea is that if you have a risk score, you should have some type of normal distribution in your population. And so a lot of what we do in medicine is guideline based. It's based on, the American Heart Association gets together and tells us what's the healthy threshold for blood pressure and how we need to treat heart failure and all these things based on these big clinical trials and studies. And so that kind of algorithm-based approach is what allows us to treat people kind of in this precision medicine framework. Because I'll just be honest with you, we can't memorize everything in medicine. And so if we have tools that help us target people who are at much higher risk, we can say, okay, for this person over here who's at high risk, maybe I see them in 3 months after this, as opposed to 6 months or a year from now. Maybe I start them on this medication sooner, as opposed to just letting them kind of have some more diet and exercise regimen alone, and then kind of check on them in 6 months. And so that's the idea of how we can use these tools. And so for this methylation risk score, we did what's called a candidate approach, and I'll talk a little bit more about that in a second. But as I stated kind of earlier, your polygenic risk score pipelines are really well developed. Essentially, you take some GWAS summary statistics that has, 100,000, 200,000, even 1 million people in it, and you kind of plug it into this program and you set your own parameters. And then using that program, it'll help you pick out what are the best SNPs or variants to include in that polygenic risk score. those types of pipelines don't yet exist for the methylation risk scores. And so we kind of had to manually attempt to duplicate that process as much as possible. So what I did was a literature review of published EWAS for kidney traits. So that's CKD, that's GFR, that's proteinuria, or the amount of protein in your urine, which can tell more about your kidney function. And so I basically said, okay, of all the top CPG sites that are coming up in these EWAS, I'm going to basically put them in a pool. All right. And then from there, how do I select which are the best ones to create our methylation risk score? And so we had what's called a target data set, which we call the Hypergen study. The Hypergen study is a family study of hypertension and genetic risk factors for hypertension, but we also have methylation data for those individuals. And so we did an EWAS in Hypergen, and we had very loose criteria. We said, if the association of in Hypergen with whatever that trait is, CKD, GFR, proteinuria, is in the same direction as it is in this published EWAS, then it makes it to the next stage of filtering. And then if that p-value is, less than 0.1, because we had very low thresholds, it'll make it to the filtering. But then also, if we have something that's what we call a Mendelian randomization CPG, these were CPGs that had found to causally influence kidney function, we said, regardless, that's coming to the next stage. And so from there, based on kind of what made it to that next stage, I kind of began playing around with different combinations of the CPG sites based on statistical significance, if it showed up across multiple studies, if there was some kind of biological plausibility. And so I was kind of picking things that I thought would be good to go into the risk score. And we'll talk about that approach versus other approaches kind of towards the end of the talk. And so from there, created our risk score, and then we tested it and we validated it in an external data set. And I'll talk about that in just a second. So these were our cohorts here. Really kind of one of the limitations of this study was that, unfortunately, having severe kidney disease is actually an exclusion criterion for a lot of these NHLBI cohorts, because having severe kidney disease is a risk factor for cardiovascular disease and death. We didn't want to have, a lot of these parent studies didn't want to have that as a competing interest or a competing factor for CVD. So really had fairly low rates of CVD, but these were the data that we had at the time. And I'll talk again about that towards the end. And so we had our Hypergen as our kind of training cohort. And then we had participants from the Golden study, which is a lipid study, the Jackson Heart study, which is the largest study of cardiovascular disease in African Americans, the multi-ethnic study of atherosclerosis, or MESA, and then different sub-cohorts of the Women's Health Initiative, as well as our validation studies. And so here you see just the EWAS done in Hypergen. And again, as the previous table showed, that there are only about 600 folks in Hypergen who have methylation data. And so we saw that if you've seen a Manhattan plot before, the way that it works is that for each CPG site, or each dot here represents an association p-value for that CPG. So say the CPG is located on chromosome 1 and it's got a p-value of 10 to the minus 4, it's like roughly here, right? And so what we do is the lower the p-value, the higher it is on the y-axis. And so as you can see here, really don't have a ton of statistically significant p-values really above this red line. So that was why we use those kind of softer thresholds just to kind of make it through that first selection criteria when we created the EWAS. And so this is actually what that methylation score looks like. So this is the weight of for that particular CPG site. In our case, that was the effect estimate from the literature review that published EWAS effect estimate multiplied by the percent methylation at that CPG site. So 0 to 1 was basically that number. This is a little bit different than the polygenic risk scores, because there's similarly a weight, but instead of this being a continuous number here, you have 0, 1, or 2 copies of that allele. So this is just a percentage. And so we take the weighted sum, we add up all our CPGs together, and that's our methylation risk score. So I think my image is a little bit off, but basically this is the kind of components of the final risk score that work the best out of all the different combinations that I tried. And really the main thing for you to see here is that a lot of these are really associated with transcription factors or different functions that, again, kind of made biological biological sense to be playing a role in turning genes on and off. And a few of these CPG sites, and the ones that have an asterisk next to them, where the causal CPG sites are associated with lupus, which we know can cause kind of downstream kidney damage, some diabetes traits as well, which also can be associated with kidney damage as well. So it says, okay, these kind of functionally make sense. So once we had our risk score, the first thing I do is actually make sure that it works. So in our Hypergen cohort, which was our test cohort, the first thing we want to see is just what is the distribution of the risk score among people who have CKD versus those who don't. And so you see here in the red, these are the folks who have CKD, and we see kind of a right shift of that risk score saying that people who have disease, they have higher risk scores. The same thing is represented in this box plot here. People who have CKD have higher scores than folks who don't. So we say, okay, that's a good thing. The next thing we want to do is say, well, we know that we just see this distribution, but are we actually predicting CKD? And so we fit these logistic mixed models, and we looked at kind of a couple of things. So we looked at our methylation score as what we call a continuous predictor. So we said a 1 standard deviation increase after we account for all of our covariates in our risk score is associated with about a 5-fold greater odds of having CKD at baseline in our cohort. And then the other thing we looked at was different thresholds of the score. And so you can see that these error bars are quite wide because as we're binning, we're getting smaller and smaller in our cohorts. But essentially, we compared the top 3% of folks with a risk score compared to the bottom 97% of folks with a risk score. And we see that they had about a 16-fold odds of having CKD compared to those who didn't. Same thing for the top 5%, about 11-fold odds ratio, top 10%, et cetera. And so we're seeing the higher your risk score, the higher your risk, even after we account for those covariates. So we say, okay, we know the score works in Hypergen, hooray. But again, I kind of rigged the score a little bit to work well in Hypergen. So we got to make sure that it works well in not just the Hypergen cohort. So that's where we went to our validation cohorts, Golden, Mesa, Jackson Hart, and Women's Health Initiative. And so again, like I showed you in that earlier table, we didn't have really high prevalence of CKD in our cohorts, but still our effect estimates showed that generally having a higher risk score after accounting for covariates was associated with higher odds of having CKD. And then after we took the kind of study-specific analysis, we did what's called a meta-analysis, and we pooled those estimates together, and we did have a significant estimate in our meta-analysis, such that in the kind of big set of about 6,000 folks, a 1 standard deviation increase in our methylation risk score was associated with a 1.56 greater odds of having CKD. The right panel here is showing you GFR. So as I said, GFR is a measurement of kidney function. The lower the GFR, the worse the kidney function, the higher the kidney disease. And so we see here again, increasing risk scores are associated with lower GFR or decreases in GFR. So again, it's working in the way that we would expect it to work if it's actually predicting our trait. So you say, okay, that's great. But also, remember, there's this whole component of methylation in the environment, and I'm really curious about these kind of psychosocial factors and how they're involved in health. And so we had some access to psychosocial data in the Jackson Hart study, and so we just said, let's just do a quick and dirty association analysis looking at the methylation risk score, CPG sites, and some factors that have been associated with health and disease. And so we looked at alcohol use, physical activity, whether or not people had health insurance, their mental health status, and even their neighborhood level factors. And we saw that for quite a few of those CPG sites that were included in our methylation score, there were some significant associations between those factors. Now, again, we would have to do what's called a mediation analysis to kind of tease out some of the directions of those relationships. And this was just kind of a quick association just because we were curious, but that is potentially a future project. And so just to kind of summarize, this was my dissertation work, that in this methylation risk score, we were able to predict CKD in multiple cohorts. I didn't show this in the slides, but we did all these sensitivity analysis that accounted for major risk factors for CKD, hypertension, diabetes, smoking, and obesity. And we still saw that our methylation score was a significant predictor. So it's adding information beyond what we already know about risk factors for CKD. The other thing too is that some of those CPG sites that are included in our methylation risk score, they're also associated with physical activity, with mental health, with neighborhood features as well, and may give us some information about linking the environment to methylation to kidney disease. And that is a potential other source of study. However, there were a few limitations. So although our sample size was over 6,000, our training data was about 600 folks. And that was just kind of how it worked as far as the approvals that we got for the data and the way we could combine the data. Additionally, methylation cohorts tend to be much smaller than genomic cohorts that are in the hundreds of thousands to millions. And so we already know kind of from the beginning, we won't have as much power as a big genetic study, but we still were able to kind of gather as much data as we could. The other thing too was that because of the data that we had, particularly with Hypergen, it was based on what's called a 450K methylation array. So this is of our data that were available, we had about 450,000 CPG sites that we could test it and kind of select among for this methylation risk score. However, there are newer arrays that have since come out that are in other studies that have 800,000, 900,000 CPG sites. And so we didn't get, as many CPGs as we could have used for that. And so potentially increasing or using new array data could improve our risk prediction. And then finally, kind of like I told you before, really the methylation risk score method space is not as robust as the polygenic risk score space. And so the approach that I used, which was kind of what we had at the time to work with, is probably a little bit more susceptible to bias than a hypothesis-free approach. And so that kind of leads into some future directions of some things that we're kind of currently working on. So towards the end of my PhD, I wanted to really look at the clinical utility of these polygenic and methylation risk scores. Now, to be honest, part of it was I had to go back to med school by a certain window or else I would have to wait another year. So I had to cut some of these analyses a little short. But essentially, one of the things we quickly looked at was whether or not these methylation risk scores could predict kidney failure. Because if you're thinking about a doctor in a clinical setting, and my apologies, I think the image is slightly off, but I don't need to use some special score to tell me if a person already has disease. I can tell that based on my physical exam, based on my lab findings. I need these risk scores to tell me who's going to get worse disease or who's going to progress to kidney failure. And so because we had cross-sectional data, we developed all these risk scores based on who had disease at the time. Now we want to figure out who's going to get kidney failure at the end. And so we compared our methylation risk score to the kidney failure risk equation, which is that first row. This is the gold standard clinical risk score for predicting who's going to progress to kidney failure within a certain time point among people who have chronic kidney disease. And so as we expected, the clinical risk score was going to be your best solo predictor. However, the methylation risk score still was a significant predictor of kidney failure. So this is essentially representing folks who were in the top 5% of the methylation risk score compared to people who were in the bottom 95% had about a 3.5-fold higher risk of developing kidney failure compared to, again, to those who were in the bottom 95%. And we saw that our area of the curve was modest as well in our full model, not as strong when we only use the methylation score as a solo predictor. But really, one of the things that we saw was that when we started adding the clinical risk score and our polygenic and methylation risk scores together, we got some improved prediction, not for the entire cohort per se, which is why these AUCs kind of look about the same, but really, we saw that for people who were already at high clinical risk, we were able to further refine some of that risk stratification when we included the methylation score, when we included the polygenic score. And so for us, like I said, we developed this methylation score. It predicted incident outcomes, and that actually combining that score with the clinical score actually improved our risk, particularly for people who were already by that clinical score defined as being at high risk. We also did some interaction modeling and saw a significant positive multiplicative interaction between the clinical risk score and the methylation risk score. And so this was a really exciting work. However, we still had those limitations regarding the actual methylation score that we had. So kind of what I'm working on in this mini postdoc is refining that approach to the methylation risk score. So like I said, we know that there are these great methods for the polygenic risk scores that are hypothesis-free and they work really well. But the question is, can they actually be applied to the methylation risk score? So I'm going to be taking two different methods that are, quote, hypothesis-free approaches. So as opposed to me kind of cherry picking what I think is the best, I'm letting the statistics kind of guide the selection of those best CPG sites and saying, okay, can they predict better than the approach that I used previously in my dissertation? The other thing we're going to do is use just more data. So we're going to have a bigger array that's going to have 800 plus thousand CPG sites. And we're also going to use summary statistics from a recently published EWAS that had about 40,000 people in the meta-analyzed set. And so the idea is that we'll have way more power, we'll have way more CPG sites, and we're going to use this hypothesis-free approach. And we're going to see if that works better than our previous score. And then see, hey, can we actually still predict incident kidney failure well, and kind of look at some of the similar things that we use in my methylation score that we created in my PhD. And so just to kind of summarize, I don't have any results for this yet. I will say we're waiting on final approval, so fingers crossed. But to summarize that, CKD is a major risk factor for cardiovascular disease and is a leading cause of death globally. These epigenetic modifications like DNA methylation can be useful for predicting CKD and kidney failure, but we still got to refine our methods a little bit more. And yeah, you should all be epidemiologists for those of us in the room who are not yet epidemiologists. So with that, I just want to thank the funding, the participants of all the various studies and their data that I use, and as well as the Irvin Lab as well. So I know that was short, but just wanted to kind of get through and share my words.